So let's jump to the basics of the RNA-seq protocol.
So here on this slide, I'll illustrate the basic RNA sequencing protocol. It's worth noting that RNA-seq has been going on for well over a decade now. And so there's actually many different RNA-seq protocols for many different scenarios, like if you're working with FFP samples versus just primary tissues. There's many different protocols for all of these different scenarios. But we'll just kind of talk about a vanilla RNA-seq protocol because a lot of the other protocols share a lot of similarities with this one. And so to get some of the terminology down, basically when I say transcriptome, I'm referring to basically all of the different RNAs that are being produced in the cell. And so this includes your classic kind of poly-A-tailed mRNA, but also includes a lot of non-coding RNAs that we'll talk a little bit about in a few slides. And so one of the most important distinctions here is that the transcriptome refers to all RNA products that are actually being expressed in a cell. And so this does not include all of the, for example, possible isoforms that might be produced from a gene with multiple introns and exons, but it really only focuses on the things that are actually being produced in a bunch of cells. And so the way that RNA-seq protocols typically start out is that you typically start out with isolation of total RNA, so that's basically any RNA being produced by the cell. And then you actually typically perform a selection step. And so the selection step is shown on the next slide. It's not shown here. But basically the idea is that most of the RNA in a given cell is made up of ribosomal RNA and RNA from mitochondrial genes. And so if your focus is, for example, poly-A-tailed mRNA, that actually occupies a very small proportion of the total RNA in the cell. And so if you want to just sequence poly-A-tailed mRNA, then you need to actually perform some selection for that. But once you've performed selection for your given target type of RNA, then you typically fragment your long transcripts into shorter fragments. And then what you do is because you can't sequence RNA directly, sequencing technologies are generally focused towards sequencing DNA. So you have to convert your RNA to DNA. And so you do so by basically annealing some random oligos to your RNA fragments, and then you extend them using reverse transcriptase. So that produces basically hybrid double-stranded molecules of both DNA and RNA. So at that point, you then add RNA-seq to get rid of the RNA, and then you add DNA pull 1 plus some primers to basically produce double-stranded DNA molecules or double-stranded cDNA. And at that point, you then do some further size selection to select reads or fragments of a specific length. You then do sequencing and then basically map those reads to the genome. And so because you're sequencing RNA and basically only a small fraction of the genome is typically transcribed, then most of your reads will fall to a small regions across the genome. And so we'll speak about this a little bit later, but you can either align reads back to the original genome if you don't know where the transcribed regions are, or you can map them to, for example, databases of just expressed regions of the genome, or even what we'll talk about later, what are called splice junction databases. But the idea is that, for example, if you map them back to the original genome, then in principle, the reads will typically align to, say, for example, exons of protein coding genes. And the idea of RNA-seq is that, roughly speaking, the number of reads that map to a given gene is roughly proportional to how often that gene was transcribed in the cells that you sequenced. And so that's not entirely true because, for example, genes that are longer in length and have more base pairs tend to have a higher number of reads, even if the number of transcripts is the same. But we'll get into that detail later. But the idea is that you can sequence reads, for example, map them back to the genome to figure out how often each transcript is expressed. And then in a differential gene expression test, for example, you could just compare the number of reads that map to a given gene across different samples from either cases and controls, and then test whether or not a given gene is significantly differently expressed in your cases versus your controls. And so I mentioned on the previous slide that there's typically an RNA selection step where you take your total RNA and potentially subset that RNA into a more focused group of RNA that you're typically interested in. And so the way this typically works is that, again, your RNA-seq strategy starts out by starting with a collection of cells, either from like a tissue or say that are dissociated from the blood. And you then extract out your RNA, and then you perform selection. Right, and so on one end of the spectrum, if you don't perform any selection, you can basically sequence total RNA, which again is vastly composed of ribosomal RNA and RNA from mitochondrial genes. And on the other end of the spectrum, you can select for example, only poly-A-tailed mRNA, and then just sequence basically protein coding genes. And so the choice of what you select for is driven in part based on your experimental design, because, for example, you might just care about poly-A-tailed mRNA, or you might be focusing specifically on any kind of express transcript, like non-coding RNAs, for example, that don't have poly-A-tails. On the other hand, there's also some practical considerations. So, for example, for samples that are fixed, right? So those are FFP samples, for example. A lot of times, FFP basically causes a large degradation in RNA in your samples. And so oftentimes, you can't actually just do general poly selection because your RNA is just too degraded. And so oftentimes then you might switch to a selection strategy like doing exome capture, which is more targeted and can therefore more easily pick up specific targeted genes that you're interested in. And so again, when you do total RNA selection, that's basically just like not selecting at all. And again, in most species, ribosomal RNA makes up as much as like 95 to 98% of all the RNA molecules in a cell. And so for that reason, it's not that common for people to just sequence total RNA without doing some kind of selection or enrichment. The next kind of least selecting procedure is what's called ribosomal RNA reduction. And so here the idea is that you're trying to specifically deplete the ribosomal RNAs by hybridizing those ribosomal RNAs to specific oligonucleotides. And so the idea is that when you kind of capture these ribosomal RNAs and immobilize them, then basically all your other RNA molecules can be basically washed through and used as input to RNA-seq library construction. And so again, depleting ribosomal RNA means that you're still left with a lot of RNAs that are going to map to, for example, mitochondrial genes, and you're also left with a lot of non-coding RNA that are not ribosomal, for example. So in contrast to total RNA or the ribosomal RNA reduction strategies, poly selection is relatively specific. And so here the idea is that the vast majority of protein coding genes have poly-A-tails on their mRNA. And so you can select for poly-A-tailed mRNA specifically by using, for example, beads that have like poly-T oligos hanging off of them. And so what will happen is that the poly-A-tails will anneal, they will hybridize to the poly-T tails hanging off of these beads, and you can essentially select out these poly-A-tailed mRNA, and then only sequence those poly-A-tailed mRNA. And in doing so, you're essentially selecting just for that one species of RNA, and getting rid of both your ribosomal RNA as well as your non-coding RNA that are not poly-A-tailed, as well as most of the other non-coding RNA. So the final selection strategy is called cDNA capture. And the idea here is that you're capturing RNA from very specific targeted regions across the genome by, for example, designing arrays that have complementary probes to specific exons of protein coding genes, which will then capture RNA from those regions that you can then pull down and sequence. And so this strategy is used more often if you're trying to sequence genes, for example, that are very rarely expressed. And so if you didn't select for them, they account for such a small proportion of RNA in your cells that you basically never sequence them. And so another application of cDNA capture is basically when you have like a lot of heavy degradation of RNA from fixed samples. And so in those cases, this approach is basically more sensitive to picking up transcripts than general poly-A RNA sequencing. But again, in general, cDNA capture is less frequently used, mainly because sequencing is getting more and more cheap, and our processing of even samples like fixed samples is getting better and better, and so it's typically not as necessary to have to capture and target specific transcripts unless there is one specific or small set of transcripts that you really need to quantify very accurately. So oftentimes in practice, it's pretty hard to get consistently good RNA across all of the samples in your study. And so, for example, for people who try to study gene expression patterns across multiple human tissues, oftentimes your tissues are coming from a variety of individuals. Some individuals are still living when they donate their tissues, whereas for some types of tissues like brain samples, you can only typically get those from deceased individuals. And so RNA quality in general degrades quickly as an individual, once an individual has passed away. And so the quality of your RNA can vary widely across your samples. And so the RNA integrity number is a pretty common quality control metric that indirectly measures how much degradation in RNA has occurred in each one of your samples. And so the idea is that basically you run a gel on a subset of your total RNA from your sample, and depending on how much degradation in RNA has occurred, then you basically expect to see different kinds of banding patterns in your gel. And so, for example, if you have not done any RNA selection and you're just looking at total RNA, then you'd expect some pretty big bands that correspond to the 18S and 28S subunit of the ribosome. And furthermore, you should see a pretty high ratio between the 28S band and the 18S band. And that typically indicates little degradation. And so RNA integrity number, or RIN, of 10 is basically the best RIN value that you can get. And you can see that in this gel for a RIN of 10, you can see that your 28S band, or your peak, is much higher than the 18S peak. And so the little peak that you see on the left-hand side corresponds to basically a 35 base pair marker that you typically spike in at a fixed concentration for calibration. And so in contrast, if you have a sample that exhibits like a RIN of 6, then basically you'll still see kind of large peaks at the 18S and 28S regions, but the 18S peak might actually be bigger than 28S peak, which in this case indicates a little bit of degradation. So as the RIN number decreases, you can basically see a higher frequency of smaller fragments. So that essentially indicates heavier and heavier degradation. And so, for example, at a RIN of 2, you basically can't see any 18S or 28S peaks, which, again, if you're looking at total RNA samples, is what should be composing the bulk of your RNA. And so RIN of 2, for example, it would just tell you that your sample is pretty much garbage. And similarly for RIN of 3, there's just like a ton of noise in your total RNA sample, and it probably indicates that you should just go and collect another sample. And so I'll just briefly mention PCR here. So particularly when it comes to bulk RNA sequencing, PCR is again one of those kind of necessary evils that still gets used frequently, in particular for bulk assays. And so when we get to the single-cell RNA-seq assay, for example, we'll talk a little bit about how some of the problems with PCR have been mitigated. But here again, just for illustration here, PCR is an amplification procedure where you can start with a relatively small set of double-stranded DNA molecules, and then through the addition of, for example, random primers, well, first you have to denature your double-stranded DNA molecules, randomly anneal different primers, elongate, and basically repeat this procedure. And in doing so, basically after like K cycles of PCR, you should basically increase, in theory, the number of double-stranded DNA molecules by 2 to the K times. And so, for example, after one round of PCR, you should double, in theory, the number of molecules you have. And then after 2 rounds, you should essentially quadruple the number of double-stranded DNA molecules you have. And so again, as you've probably learned many times at this point, PCR is a non-perfect process. And so oftentimes it leads to artifacts because certain sequences tend to get amplified much more frequently than others. And so although everyone knows that PCR leads to an increase in bias in the representation of certain DNA fragments compared to others, it's oftentimes necessary, specifically for RNA sequencing, for example, because oftentimes if you're working with certain types of samples like FFPE samples, formalin fixed samples, then you have very little RNA to begin with. And so a lot of these bulk assays need RNA from millions of cells. And so you need to use PCR in order to generate enough RNA, or in this case, cDNA molecules, in order to properly sequence all the RNA in your samples. And so bulk RNA assays have a lot of challenges associated with them. And so, for example, one of the biggest challenges associated with bulk RNA sequencing is essentially sample heterogeneity. And so here again, the idea is that in your bulk RNA assay, you have to collect RNA from like millions of cells in order to get enough RNA to do the sequencing. And so when you're working with primary tissues, for example, it's rare or impossible to find a tissue or a million cells that are all identical. And so typically there's multiple cell populations present. And so it's worth pointing out that this is true even when you do things like FACS sorting to sort out cells that express certain cell surface markers, because there is no such thing as there basically is no such thing as a set of cell markers that uniquely identify exactly one cell population and doesn't capture any other type of cell. Basically, you should assume that whenever you're doing bulk assays, there's always multiple distinct cell populations present, and the expression, the transcripts that you sequence are from a collection of distinct cell types. And so this is a problem because, for example, if you are trying to compare how gene expression varies across samples, then what you're actually measuring is both a potential change in gene expression within each cell population, as well as you could also be picking up just changes in proportions of these different cell populations. And so essentially, population heterogeneity or sample heterogeneity is essentially a confounding factor in gene expression analyses. Another potential problem you have is one that we talked about with ChIP-seq. So for example, you might have multi-mapping reads. So reads, especially if you're doing short read sequencing, might map to multiple regions in the genome. And this happens pretty frequently. And so because it happens so frequently, how you quantify transcript expression using these multi-map reads can basically, your quantifications can change significantly depending on how you deal with those multi-mapping reads. Also, alternative splicing turns out to be a pretty big problem because currently the vast majority of RNA sequencing experiments are still done with short read sequencing. And so alternative splicing is difficult because, especially if you have genes with a large number of exons, it can be hard to quantify, for example, individual isoform level expression because your reads are short. And so at most your reads might only span one junction. And so it's impossible to distinguish expression of isoforms from genes with many, many, many exons. Furthermore, it can, depending on whether those isoforms have already been characterized. So depending on whether someone's been able to distinguish which potential isoforms are not expressed versus those that are expressed, then you may not be possible to even identify which splice junctions are actually being expressed, never mind entire isoforms. And so another classic problem of RNA sequencing is that you can have a huge range of gene expression patterns across the different genes in your cell. And so, for example, ribosomal genes and mitochondrial genes are typically some of the most highly expressed genes in any given individual cell. And so they tend to dominate the reads that you sequence in RNA sequencing experiments. So even when you deplete ribosomal RNA, for example, oftentimes you don't deplete mitochondrial genes, and so our mitochondrial genes essentially soak up most of the reads that you sequence. And so that essentially makes it harder to detect expression of more poorly expressed genes. Some other problems involve measuring expression of different types of RNA. And so historically, a lot of people have focused on expression of protein coding genes, but we now know that non-coding RNAs are actually more numerous than protein coding genes. And so it can be actually hard to capture small RNAs. And so typically you might have to perform multiple selection procedures in order to sequence, say, poly-RNA versus small RNA, and so on and so on. And finally, again, as I mentioned with the RNA integrity slide, it's typically hard to get really high quality RNA across many samples. And so when you're comparing transcripts and gene expression patterns across multiple samples that you might collect, RNA integrity and diversity, library complexity essentially, can change between samples, and so this can affect how accurate your comparisons between samples are.